For inviting me to the UBC AMDB seminar series. I think it's an excellent initiative, and I'm really happy to be here. And today I will talk a bit about the effect of nitrate on the oral microbiota and how this could affect oral and cardiovascular health. Okay, so a short introduction to the oral microbiota. These are all microorganisms that reside inside the oral cavity, and they can be found on all oral surfaces, such as the tongue, the teeth, the gingival crevice, and the inside of the cheeks, where they form biofilms, these structured communities. The main inhabitants are bacteria, but there are also fungi, protozoa, and archaea. And on the tooth surface, we have to apply oral hygiene to keep the surface clean, as mature oral biofilms are associated with oral diseases and especially with the modern diet. Bacteria from these surfaces disperse out of their biofilms into the saliva, leading to a high number of bacterial cells per milliliter of saliva, namely between 100 million and a billion colony-forming units per milliliter. And over here we kind of have the classical representation of a biofilm like we used to see them in biology books, but now thanks to new technology that is named Classy Fish, we can get these nice pictures of oral biofilms, and this is how they actually can look. So here we have dental plaque, and here we have the tongue biofilm. This is work by Jessica Mark Welch's group in Boston. And over here, there's one human tongue cell, and around that, there's a biofilm. All these little circles are individual bacterial cells. That's why there are so many, because they're much smaller than human cells. And each color is a different type of bacterium. And they kind of form colonies because they are dividing. So just like on an agar plate, they form colonies, they do as well in the biofilm, which is logical, but we had to see it to believe it. And now we'll go into more detail about the dental biofilm. So based on these images, the authors designed this model where there's like this long bacterium called Corynebacterium that's forming the pillar of the dental plaque. And it starts at the tooth surface and ends at the very end. And unlike what we always believed was true, these streptococci are mainly at the outside of the biofilm, where they kind of form a shield for anaerobic bacteria as the streptococci are consuming the oxygen, so that anaerobic bacteria can grow inside the biofilm. And all these different species, they work together to degrade complex host molecules such as glycoproteins in the saliva. So they kind of help each other to grow and form this very diverse community. And they're also our friends in health, at least, because one function that the oral microbiota has, like the other microbiota on human body surfaces, is that it makes it more difficult for external pathogen to colonize because the microbiota occupies the surfaces, produces antimicrobial molecules, and contributes to immune system development and maintenance. Additionally, while we know that the gut microbiota is very important for systemic health, now we also know that the oral microbiota contributes to that. Specifically, it has been shown that the oral microbiota can contribute to vasodilation, blood pressure regulation, metabolic health, and exercise performance. And this is all mainly, as far as we know now at least, because of nitrate metabolism. And we obtain nitrates from our diet, mainly from vegetables. It has been estimated that we obtain over 80% of dietary nitrate from vegetables. And oral bacteria can convert this nitrate into nitrite and further into nitric oxide. And nitric oxide really is the magical molecule, as it is a vasodilator, a metabolic regulator, an immune regulator, and an antimicrobial molecule. And it has been shown that nitric oxide kill certain bacteria associated with periodontitis, such as Porphyromonas, Fusobacterium, and Prevotella. And when we eat vegetables, the bacteria that use nitrate, which are health associated, including Rothia and Neisseria, they increase in number. And apart from getting nitrate from vegetables, the human body itself also produces endogenous nitrate because we have enzymes that can produce nitric oxide from arginine. And under certain conditions of the body, this nitric oxide can oxidize into nitrates. And then, even when we're fasting, this pathway becomes important to recycle part of this endogenous nitrate back to nitric oxide. So this is a function of the oral microbiota that's also important when we're not consuming nitrate-rich vegetables. But oh yeah, and while we strongly depend on bacteria to reduce nitrate to nitrite, this is something the human body cannot do effectively. So we do depend on our oral microbiota for this step. We can, however, convert this nitrite into nitric oxide, as nitrite reacts with hemoglobin and with the acid in our stomach forming nitric oxide. So this way we can have a nice symbiosis with the oral microbiota that produces nitric oxide that we then convert into, that produces nitrite that we then convert into nitric oxide. And let's have a look now at what happens exactly when we consume nitrate-rich vegetables. This is a figure that I always include in my talks. When we consume nitrate-rich vegetables, we swallow the nitrate and then it gets into our bloodstream. So far, this is quite common for nutrients. But what's really special about nitrate and really unique about nitrate, that we don't only have it in the mouth while we chew on vegetables or while we drink the beetroot juice and it touches the surfaces. We swallow it, and then part of it, around 25%, goes back to the mouth because the salivary glands take out plasma nitrate from the blood and concentrate it into the saliva into very high levels, levels that are about 10 to 80 times higher than fasting salivary nitrate levels. And the salivary glands do this so that oral bacteria can convert this nitrate into nitrites. This is the step that we cannot do ourselves. Then we swallow the nitrite produced by oral bacteria, and there in the stomach and in the bloodstream, it reacts with acid and hemoglobin, forming nitric oxide, which is a vasodilator. So it contributes to a healthy blood pressure. And from studies with chlorhexidine, we know that if we impair the oral microbiota in a temporary way, then no more nitrite is produced. Nitric oxide levels drop, we lose part of the vasodilation, and chlorhexidine for several hours completely impairs the blood pressure lowering effects of nitrate. So we know the oral microbiota is essential in this pathway. Okay, so it's important to keep this pathway intact and not just for blood pressure. This pathway can also improve physical performance, other cardiovascular health parameters, oral health, wound healing, cognitive function, and glucose levels. So it's a very important pathway. And here's some data to support the figure that I just showed to you. In this study by Govoni and colleagues, they gave a nitrates-containing drink to two groups of individuals. One group just had the drink by itself, and the other group used chlorhexidine before the nitrate-containing drink. And these are the closed circles, the chlorhexidine group, and it seems like there's more nitrate there, so that could sound like a positive thing, but it's actually because in the chlorhexidine group, there's no nitrate reduction to nitrite in the 3 hours in which this was measured. In the plasma, the nitrate levels are similar in both group after the nitrates drink intake. But when we look at the nitrites, we can clearly see that there's much more nitrite production in the control group compared with the chlorhexidine group. And the differences between these two lines is really the power of the oral microbiota in nitrite production that directly affects nitric oxide's bioavailability. And here are two examples, or will be two examples of studies in which they gave 1 week of chlorhexidine, 2 doses per day of 0.2% chlorhexidine mouthwash. And in one study, they found a small increase in blood pressure. This is also temporary, but both systolic and diastolic blood pressure increase. So that they find this increase in some studies, not in all. There are multiple factors involved, so it must really depend on the study group and conditions. And in another study by Raúl Vescos and colleagues, they found that chlorhexidine also decreases the oral pH and increases lactic acid levels. So, as kind of a summary, chlorhexidine can be very important as it can reduce plaque accumulation and gingival inflammation, and it's recommended for short-term use after oral or periodontal surgery. So, if your dentist recommends it, please use it. But healthy individuals probably shouldn't use strong antimicrobial compounds because the oral microbiota is important for us. And repeated exposure to chlorhexidine may promote bacterial adaptation and cross resistance to antibiotics. And as we have seen so far, it can alter the oral microbiota composition and metabolic activity. So, in a for at least several hours, it impairs nitrates metabolism, can lead to a small increase in blood pressure and decrease oral pH. And let's continue now to have a look at the oral pH. Why is this important? Because to go from sugary snacks and drinks to caries, several bacteria processes happen that decrease the pH and can lead to enamel damage. Because these sugars are not only a nice snack for us, some bacteria in the mouth really enjoy them, the saccharolytic ones, and they ferment these carbohydrates into organic acids, including lactic acids, lowering the pH, which selects the acid-tolerant saccharolytic species, thereby shifting the microbiota to a karyogenic one that is better at fermenting carbohydrates. So if we keep consuming carbohydrates in a frequent manner, then the pH will keep decreasing to levels that can demineralize the enamel, and that increases caries risk. So ways to prevent this is to remove the biofilms, remove the karyogenic bacteria with oral hygiene, or use fluoride that increases enamel resistance. And we could, of course, also change our diet, which is probably the most powerful strategy, avoiding added sugars and increasing vegetables as well, because vegetables contain nitrate that can prevent acidification in several ways. So here we're back to this figure where oral bacteria reduce nitrate to nitrite. And apart from that, they also have enzymes to produce nitric oxide. So this already happens in the mouth, where nitric oxide can inhibit sensitive species. Perhaps some karyogenic species could decrease in the presence of nitric oxide. And alternatively, they can reduce nitrite to ammonium. And what's very relevant for the pH is that during these reduction processes, all these different steps, protons are being consumed. And the pH is the levels of protons in the environment. So by consuming, then you're increasing the pH and preventing acidification. And finally, nitrate-reducing bacteria tend to use lactic acid as a carbon and electron source during nitrate metabolism, forming weaker acids that could also limit acidification. And all these molecular mechanisms were quite well described in environmental microbiology, so microbiology of soil and water communities, and we wanted to see if it would also be relevant in the mouth. So what we did was we incubated saliva with nitrate or without nitrate in sugar-rich medium. And after 5 and 9 hours, we observed what happened. And over here is the nitrate that we added at the beginning to the mixture of saliva and sugar. And then over time, the nitrate had been metabolized. Most of it had been metabolized after 5 hours. And after 9 hours, there was no more nitrates detected. And the nitrite first increased and then seemed to have been metabolized as well. We also looked at ammonium in the control condition without nitrate and the nitrate condition. And in both conditions, it increased after 5 hours and it further increased after 9 hours, but at both time points, it was higher in the nitrate condition. Lactic acid or its ion, lactate, increased after 5 hours and was partly consumed after 9 hours in both conditions, but it was significantly lower in the nitrate condition at both time points. And in accordance with these nitrate metabolism steps happening that consume protons and lower levels of lactate, the pH was also higher in the nitrate condition at both time points. And this pH drop here in the control condition after 5 hours was very significant, but it was prevented in the nitrate condition because in some individuals the pH did not drop. So that's very promising, but all of this was in vitro by incubating saliva in vitro. So we wanted to see if it would also happen in a clinical study. And in this study that you can see here, we gave individuals a beetroot extract and then waited for 1 to 4 hours and then gave them a sugar rinse. And we basically saw that a single dose of nitrate increased the salivary nitrate, decreased the salivary lactate, and limited acidification. So a single dose of nitrate can already protect you against sugar. And then in another study by researchers in Scotland, who are also our collaborators in other studies, they gave a beetroot extract to individuals and then waited for 1 hour and then gave them sugar in the form of sugar-rich sport drinks. And these decreased the pH. Additionally, they combined this with 90 minutes of exercise, which also decreases the pH. And during this exercise, they consumed more sugar-rich products, which further decreased the pH. And I will show you the results of this study because I think it's a great and very complete study in the next slide. This study had 4 arms. It had a negative control where individuals consumed water before exercise and water during exercise. Then there was the nitrate condition where they consumed beetroot juice before the exercise and sugar-rich sport drinks during the exercise. The placebo condition where the nitrate was removed from the beetroot juice, and a positive control where water was consumed before exercise and sugar-containing drinks during the exercise. And these are the results. So these are the pH levels in the different groups. Here's the pre-exercise pH, where the pH drop resulted from one sugar-containing drink. And here is the post-exercise pH, where the pH drop resulted from 2 drinks and 1 gel in total containing sugar. And to make it a bit easier for everyone, I selected the nitrate condition over here and the placebo condition over here. And the area, it's already clear that the nitrate condition is higher than the placebo condition. So nitrate is an essential component of beetroot juice to buffer the pH. And And when looking at the area under the curve, the nitrate condition was statistically identical to the negative control, while the placebo condition was identical to the positive control, both before and after exercise. So they showed that nitrate can protect athletes from the sugar-containing products they use to basically survive some of their longer periods of exercise. And this could be a very promising strategy to limit caries development in this group that is at high risk of caries. Then in another study in collaboration with Constanza Fernandez in Chile, we looked at what would happen if we would mix nitrates and sugar. And specifically, we added 2 grams of sugar and 5 milligrams of nitrate to a 10 milliliter oral rinse. So there was much less nitrate than sugar. And in a cross over design, we looked at three different types of rinses, one with only nitrate, one with nitrate and sucrose, and one with only sucrose. And these are the results. So this is the pH drop in the minutes after the sugar rinse, and then there's lactic acids, nitrate, and nitrite. And we see that here's the pH drop when we only have sugar in the rinse. When we add a little bit of nitrate, the pH drop is significantly limited, and this is because a small increase in salivary nitrate and nitrites during the first 12 minutes after rinsing. And the lactic acid levels in the end are also lower in the rinse containing nitrate and sugar than in the rinse containing just sugar. So now we are wondering if we should start adding nitrate to some of these products because that could really have a big impact on the burden that caries is causing. If we could just keep the pH above 5.5, maybe we can prevent a lot of cavities. And we're looking into this possibility, for example, for sport drinks with a company. So hopefully that will lead us to somewhere. And going back to this in vitro study that I already showed to you, apart from looking at the bacterial metabolism, we also looked at the bacterial composition. And we found that Rothia and Neisseria increased as we expected from other studies. And then we saw that different genera were decreasing. And that these genera could be grouped in caries-associated, Streptococcus, Veillonella, and Oribacterium on a genus level are associated with caries, and or periodontitis and halitosis associated Porphyromonas, Fusobacterium, Brevotella, and Leptotrichia. So these results were nice, but they are again from an in vitro study, and more or less at the same time and in the years after this study, different clinical studies were done with nitrate intake, and they were summarized in two systematic reviews, one by researchers in the UK and Australia, and one to which I also contributed. And here is a summary of the results. So the clinical evidence based on 9 studies that were included in this systematic review, indicated that nitrate intake increases Rothia and Neisseria, increases the salivary pH, and can decrease gingival inflammation. So they proposed that nitrate intake could be a possible nutrition strategy to improve oral health. In our systematic review, apart from clinical studies, we also added studies with real oral samples that were incubated in vitro. And we included 12 studies, and we found that there were some consistent results, including an increase in Rothia and Neisseria, a decrease in Brevotella, Veillonella, and Streptococcus, and an increase in the salivary pH. And we concluded that future studies should evaluate the changes on a species and strain level. And most of the clinical studies that were included in these systematic reviews were studies in which individuals were consuming nitrate in the form of a vegetable juice for 1 to 6 weeks in healthy individuals. And what was remarkable was that there were no patients with periodontitis, while some of these results indicate that nitrate could be beneficial to prevent periodontitis development because Rothia and Neisseria are periodontal health associated. There was one study that showed that nitrate can decrease gingival inflammation, and also decreasing Prevotella can be beneficial as this genus is associated with periodontal diseases. And let's start looking at periodontitis development from a microbiology point of view. When dental plaque is accumulating, for example, resulting from a lack of oral hygiene, then anaerobic species increase and the human body can respond with gingival inflammation. And this will increase inflammation tolerant, proteolytic, and alkalophilic species. shifting the microbiota to a more baropathogenic one. And in gingivitis, we can already see some changes, and in periodontitis, it becomes very clear that the subgingival plaque has a different dysbiotic composition. And when having a closer look at the dysbiosis in periodontitis, we can see that certain disease-associated bacteria increase, such as Treponema and Porphyromonas. But what's equally as important is that health-associated species are decreasing in number. And this word cloud by Magda Ferrese's group shows that shows the bacteria that are periodontal health associated, and the bigger the word, the stronger the association. And many of the most health-associated bacteria are actually nitrate-reducing bacteria. So we can conclude that there are lower levels of nitrate-reducing bacteria in periodontitis. And we were wondering if, because of this, we could reduce this dysbiosis over here by adding nitrates to periodontal communities or nitrate-reducing probiotics. And to study the dysbiosis in oral samples, there's a very useful tool, which is called the subgingival microbial dysbiosis index. You can apply this to 16S sequencing data, so bacterial composition data. And what this machine learning-based index does is it detects biomarkers associated with health and disease and calculates a number that represents the amount of dysbiosis in your sample. And over here are the health-associated bacteria, and once again, around half of them are nitrate-reducing bacteria. And what the authors did, this index is by Chen, Marsh, and Al-Hebshi. What they did is they applied it to the samples of our study, to the sequences of our study that I showed you before, the in vitro study in which we added nitrate to saliva, and they found that the nitrate had dysbiosis lowering properties, which were significant after 5 hours of incubation. So that was nice, but what's much more relevant is that they also applied this index to a study by Jaco Snyder and colleagues, where they compared lettuce juice without nitrate with lettuce juice with nitrate in individuals with gingivitis. And they found that lettuce juice without nitrate did not affect the dysbiosis index, but lettuce juice with nitrate significantly reduced the dysbiosis index. And this was accompanied by a decrease in gingival inflammation and an increase in Rothia and Neisseria. So these results were promising in healthy individuals and individuals with gingivitis. And we wanted to know if nitrate could also modulate periodontal communities of individuals with periodontitis, which are already dysbiotic and have low numbers of nitrate-reducing bacteria. So to test this, we incubated subgingival plaque of 30 periodontitis patients in our in vitro system that measures real-time biofilm growth with small electrons at the bottom of wells of a 96 well plate that produce small amounts of electricity that do not affect the bacteria but can quantify the bacteria that have formed biofilms and have attached to the electrodes. And like that, without touching your sample, you can get a biofilm growth curve. And we grew these communities for 7 to 12 hours with 5 or 50 millimolar sodium nitrate. And we chose this concentration because 5 millimolar nitrate is a concentration that we can have in saliva after consuming vegetables, while 50 millimolar is an artificially high concentration that could be added to different products such as a periodontal gel. We also added a third treatment condition, which was combining the low nitrate concentration with a probiotic candidate. This was Rothia area, and I will get back to that later, but first we're going to focus on just the low nitrate and the high nitrate concentration. So this is the effect on biofilm growth. The black line over here is the growth curve of the subgingival plaque community over time. And when adding nitrates, there's a small but significant decrease in biofilm growth. This is the green line over here. And this is 5 millimolar nitrate. When adding 50 millimolar nitrates, the difference between the two lines becomes much clearer and is also significant. So this data indicates that nitrate could limit the accumulation of dysbiotic plaque, but this should be confirmed with other biofilm quantification methods. And here we looked at the bacterial composition. The low nitrate concentration decreased Porphyromonas gingivalis and a disease-associated Treponema species. And overall, there was a small but significant decrease in the dysbiosis index. When using the higher nitrate concentration, uh, several periodontitis-associated species decreased, including Porphyromonas species, Tanarella, Fusobacterium, and there was an increase of Rothia, Neisseria, and a unclassified Aggregatibacter species. And overall, there was a decrease of 63% in the dysbiosis index, which was significant. And now let's have a look what happens when we add Rothia area to the low nitrate concentration. So there's a big increase of an unclassified Rothia species, and we know that that is Rothia area because we are adding this. And then there are two types of bacteria that are mainly decreasing. The first one are periodontitis-associated bacterium, like Fusobacterium species and Selenomonas. And additionally, in this case, other nitrate-reducing bacteria like Rothia mucilaginosa are also decreasing. And that's probably because when you add a lot of certain type of bacteria, similar bacteria will be competing for nutrients and growth niches. And by decreasing different health-associated species apart from periodontitis-associated species, we did not see a significant decrease in the dysbiosis index when combining low nitrate with Rothia area. But because Rothia area is so strongly associated with the absence of inflammation, we still want to test the result in a clinical study. And one thing that Rothia area addition clearly did was increasing the nitrate reduction capacity. So this is the nitrate that we added at the beginning, and just the subgingival plaque reduced around half of the nitrate. While when adding Rothia area, when combining that with the subgingival plaque, much more nitrate was reduced and much more nitrite was produced, which is this light green bar. So Rothia area could stimulate nitrate metabolism, which is relevant for oral and systemic health. And that's another reason why we want to keep exploring this probiotic candidate in clinical studies. Okay, getting back to the dysbiosis index, here we have a summary of all studies looking at the effect of, well, not all studies, different studies looking at the effect of nitrate on oral communities. And here at the top, we have studies giving vegetable juices to individuals in a clinical setting. And here at the bottom, we have studies in which oral samples were incubated with nitrates. And there are different samples over here. Nitrate intake appears to lower the dysbiosis index of subgingival communities and perhaps of tongue communities as well, but this should be confirmed in a larger population, while the effect on saliva communities was not that clear. When incubating saliva with nitrate, however, there were dysbiosis lowering properties, and also when incubating subgingival plaque with nitrate, there was a decrease in the dysbiosis index. So in summary, we can say that there is some evidence that shows that nitrate can improve the microbial composition of subgingival plaque and reduce gingival inflammation. And we recently dived into literature to find molecular mechanisms behind this prebiotic effect of nitrate. Basically, we try to answer the question, how can nitrate reduce inflammation? And once again, it's mostly because of nitric oxide production. in this little bit complicated graph, or perhaps difficult to follow, but I will go over it. Oral bacteria reduce nitrate to nitrite, and they can also further reduce nitrite to nitric oxide. Um, Nitrate can also react with heme inside the body and form nitric oxide. And to go from nitrate to nitrites, antioxidants stimulate this pathway like vitamin C, while reactive oxygen species limit it and decrease nitric oxide's availability. But if we manage to keep the nitric oxide levels in a healthy physiological amount, then they bind SGC and activate cGMP signaling. And they do this, nitric oxide does this by binding in the heme of SGC. So thereby it activates cGMP signaling, which has human downstream effects. It leads to smooth muscle relaxation, vasodilation, and anti-inflammatory effects in human cells. Well, on bacteria, nitric oxide has a different effect. Here it also reacts with iron, but it reacts with the iron sulfur proteins that are membrane associated. And by reacting with these iron groups, the iron is released in the cytoplasm and can damage DNA. So while human cells can benefit quite well from nitric oxide, bacterial cells, when they're sensitive to nitric oxide, they can die because of its presence. And one of the human downstream effects is the anti-inflammatory effects of cGMP. And I will show that in the next slide, what anti-inflammatory effects can be there. Once again, a quite detailed figure, but I will just go over the main observations. cGMP signaling induced by low physiological concentrations of nitric oxide can limit inflammatory mediators and can also suppress TSP1, which avoids excessive TGF-beta expression. And excessive TGF-beta expression is associated with pathological tissue remodeling. Additionally, this pathway can increase anti-inflammatory IL-10 expression. And overall, you're basically helping the immune system to go to a more anti-inflammatory phenotype with the activity of nitric oxides. For example, the in vitro polarization of macrophages towards an M1 type is limited by nitric oxide. So the pro-inflammatory macrophage phenotype is limited by the presence of nitric oxide. And apart from having effects inside the oral cavity, periodontitis is also associated with different systemic diseases. It can increase the risk of different systemic conditions, and this has been classically attributed to parapathogenic bacteria and their products reaching different parts of the body and causing complications there, or inflammatory cytokines produced in the gums that can contribute to systemic inflammation. But when having a closer look at some of the conditions associated with periodontitis, we notice that a lot of these diseases are also associated with a deficit in nitric oxide availability. So we thought that perhaps an additional molecular mechanism explaining the link between periodontitis and systemic diseases could be the loss of nitrate-reducing bacteria, and therefore potentially losing the nitrate reduction capacity of the oral microbiota. And to see if this could be true, we collaborated with two universities in Scotland, and we incubated saliva of periodontitis patients before and after treatment, and we incubated that with nitrate to see how much of this molecule would be reduced. and we compared that with saliva of healthy individuals. And these are the results. So healthy individuals, as is known in literature, can reduce nitrate. So when you get their saliva and incubate it with nitrate, it is reduced over time. And in the healthy group, this was significant over a period of 3 hours. But in the periodontitis group, the nitrate reduction was not significant. And however, this was before treatment. After treatment, the levels of nitrate reduction were recovered to similar levels as observed in health. And in this study, we also found that after periodontal treatment, there was an increase in nitrate-reducing bacteria in subgingival plaque. So this in vitro test was nice, and based on that, we decided to start another study to see if the loss of nitrate reduction in periodontitis could have any systemic effects. And we did this by giving beetroot juice to the same 3 groups of individuals. So we have periodontitis before and after treatment, and we have healthy individuals. And we measured their blood pressure before and after beetroot juice intake. And we published the results of this study last year, and the title is quite informative. The blood pressure lowering effect of beetroot juice is impaired in periodontitis and recovered after periodontal treatment. And here are the results supporting this title. In healthy individuals, a high dose of beetroot juice lowers both systolic and diastolic blood pressure. However, this was not observed in the periodontitis patients before treatment. After treatment, the blood pressure lowering effect of beetroot juice had been recovered, and the heart rate did not change and should not change. So that was a good control. But basically, these results indicate that periodontitis can limit some benefits of vegetables, and periodontal treatment is important to keep the nitrate reduction function of the oral microbiota intact in individuals with this disease. In this study, we also looked at the bacterial composition of subgingival plaque and the tongue coating. And to make it easier for everyone, we grouped the bacteria in nitrate reducers, disease-associated bacteria, and a subgroup of disease-associated bacteria, the red complex. And we saw that nitrate-reducing bacteria in subgingival plaque were health-associated and increased in number after periodontal treatment. In the case of disease-associated species, they were periodontitis-associated and decreased after treatment as expected. Then we looked at the tongue samples and saw that the nitrate-reducing bacteria were health-associated, but they did not increase after periodontal treatment. The disease-associated bacteria followed a similar pattern on the tongue as in subgingival plaque, being higher in periodontitis and decreasing after periodontal treatment. And this indicates that there could be some exchange between bacteria of these two communities. What's important to note is that periodontal treatment did not include tongue cleaning, and we are now considering if periodontal treatment may benefit from tongue cleaning. We're checking this in a clinical study in Turkey right now because we think that with mechanical removal, just as in subgingival plaque over here, we can boost this green bar over here and increase nitrate-reducing bacteria on the tongue, which could be beneficial for the whole oral ecosystem by increasing nitric oxide availability. So hopefully we can talk more about that next time. And these results made us wonder if we could improve the clinical outcome of periodontal treatment with nitrate-rich vegetables. So we set up another study with the group in Mexico that also collaborated with us in the previous study. And this study is ongoing, so it's not complete yet, not published yet, but I can share you at least some of the methods and one preliminary result. In this study, basically, we have two groups of individuals that receive periodontal treatment. One group continues with their normal diet as usually, and the other group has a dietary intervention with nitrate-rich vegetables, providing 350 milligrams of nitrate, extra nitrate per day. And really did a great job in Mexico. They went door to door to give free vegetables to the participants every week, and they applied different motivational strategies and even gave them recipe books with QR codes with videos of quick, easy ways to prepare these vegetables that were adapted to the Mexican diet. And we will finalize this study in the near future, but for now I can show you this microbiota analysis where the periodontal treatment is decreasing bacterial diversity in the periodontal pocket, which is known from periodontal treatment and associated with more healthy composition. When combining the periodontal treatment with nitrate-rich vegetables, there's a more significant and clearer decrease in diversity, which would be a beneficial change. And even the tongue coating, which is not affected by the periodontal treatment, appears to be changing in some individuals because of the nitrate-rich vegetables. So this is a very interesting study, and once again, I think that somewhere this year it will be published. All right, so that brings me to my final slides with conclusions. Today we had a look at the effects of nitrate inside the oral cavity, the different metabolism that can lead to nitric oxide production that can modulate oral biofilms. Nitrate metabolism can also lead to lactic acid and protein consumption, increasing the oral pH and limiting caries development. And what I didn't discuss today is that in a similar way, nitrate-reducing bacteria can also use hydrogen sulfide as electron donor and thereby limit halitosis. So that's something we're looking into now, and it's very promising. And there will be another study that hopefully will be finalized this year. Apart from consuming vegetables with nitrates, we could also stimulate nitrate metabolism by adding nitrate to oral hygiene products. And this is something that we are exploring with a company now. And we could also use nitrate-reducing bacteria as probiotics that could potentially stimulate this metabolism. And then here is another slide with conclusions just to go over how the oral microbiota can change in different situations. So when we consume sugar, that can decrease the oral pH and change the oral microbiota and stimulate caries development. Nitrate-rich vegetables can lead to pH buffering and limit caries development. A lack of oral hygiene or genetic susceptibility can lead to plaque accumulation and inflammation, and that we call gingivitis, and long or repeated episodes of gingivitis can lead to the development of periodontitis in some individuals. And in periodontitis, there's a decrease in nitrate metabolism, an increase in blood pressure, and an increase in systemic disease risk. Then consuming vegetables is good for the oral microbiota. Health-associated bacteria increase in number, and vegetable consumption is also associated with a higher nitrate reduction capacity, a decrease in blood pressure, a decrease in gingival inflammation, a decrease in systemic disease risk, and an increase in oral pH. Then gingivitis can often be treated with oral hygiene, getting rid of the inflammation and getting a more healthy microbiome composition. And Yoko Snyder and colleagues also showed that vegetables increase this positive effect by decreasing inflammation. And periodontal treatment also leads to a more health-associated composition of the oral microbiota, and we are now looking into the combined effect of periodontal treatment and vegetable intake. And based on preliminary data, it looks like the microbiota becomes more health-associated in the periodontal pocket. Finally, correct chlorhexidine usage can impair nitrate metabolism and associated benefits, and it can also decrease the oral pH. And we now want to see if we could do something with nitrate in individuals that receive chlorhexidine to give these nitrate-reducing bacteria a boost after the chlorhexidine treatment. And we also are working on several studies in which we are exploring the effect of the nitrate reducing bacteria as probiotics to see if they can further stimulate nitrate metabolism and its benefits. There are no common oral health products now with prebiotic nitrates, but we can already start increasing our salivary nitrate in a way that's definitely recommended by healthcare authorities just by eating more vegetables. Harvard Health, for example, states that the more vegetables and the greater the variety, the better. So we can already start selecting these very healthy vegetable types. Here's the top 5 of nitrate-rich vegetables, including rucola or rockets, radishes, spinach, lettuce, Swiss chard, and beetroot. And in the study by Joko Snyder et al., they found that 3 doses of 67 milligrams of nitrate per day for a period of 2 weeks led to a more health-associated oral microbiota and decreased gingival inflammation. So that's similar to 3 small portions of nitrate-rich vegetables per day. Okay, I want to thank my team at FISABIO, including my PI, Alex Mira, for his continuous support, and also our collaborators throughout the world. I saw that Shauna was connected before at the University of Glasgow, but it has been wonderful to collaborate with her in the periodontitis studies, and also the team in Mexico, in Chile, Valencia, the University of West Scotland, and our collaborators in the US. And thank you all for your attention.